Hello students, today we will discuss about forms of RNA. In introduction, ribonucleic acid or RNA is a polymeric molecule made of ribonucleotides which consist of a ribose sugar, phosphate group and a nitrogenous base, which can be A, G, C and U. The molecules plays an essential role in cellular function. RNA molecules collectively are involved in various functions such as gene regulation, translation and decoding of genetic information from the DNA. RNA exists in a single-stranded form and can also function as a genetic information carrier in some viruses. Some of the important classes of RNA are hnRNA, mRNA, tRNA and rRNA. There are other molecules which are siRNA and miRNA and snRNAs which plays a vital role in the gene regulation and RNA processing. Let us discuss about types of RNA and their salient features. First, we will take up hnRNA or heteronuclear RNA. hnRNA stands for heterogeneous nuclear RNA. As its name suggests, the hnRNA is a term that encompasses various types and sizes of RNAs found in the eukaryotic cell nucleus. As we already know, RNAs exist in many forms and carry out a wide range of functions. The majority of hnRNAs are pre-mRNAs, which are newly synthesized mRNAs, often made up of two types of segments, exons and introns. The exon segments are joined together to produce a mature mRNA that encodes a protein. The non-coding intron segments are removed by means of splicing. In the next section, we'll discuss about mRNA. Among the different species of RNAs, it is only mRNA that are linearly related to DNA and to polypeptide chains as an intermediary. It is this species of RNA that carries encoded messages from the DNA molecule in the form of codons. It is this molecule that translate by decoding the information into polypeptide chains. We'll discuss more about general features of prokaryotic mRNAs, as we know that prokaryotic mRNAs are different from eukaryotes. In the first point, prokaryotic mRNAs are made up of a polynucleotide chain of specific sequences with a 5' to 3' polarity. Secondly, though it is linear, it can exhibit several but short secondary structures. In the third point, sizes of the mRNA is more or less proportional to the gene from which it generates. In the fourth point, the polypeptide chain it produces is directly proportional to the coding size of the mRNA. Longer the protein, longer is the mRNA and vice versa. In the fifth point, all mRNAs consist of a 5' non-coding called 5' UTR called leader sequence, whose length and sequence varies from one species of mRNA to the other. The leader sequence in every mRNA contains an all-important structure sequence called Sine-Dalgarno sequence, 7 to 9 nucleotides upstream of the start codon AUG. In the sixth point, all mRNAs also contain a non-coding 3' terminal sequence whose structure, size and function vary among the members of each species of mRNAs. In the seventh point, each functional mRNA consists of coding region which lies between 5' leader segment and the 3' terminal non-coding sequence. In the eighth point, the coding region starts from the initiator codon, also known as AUG or GUG, and ends in the terminator codon like UAG, UGA or UAA. In the next point, The region between initiator codon and terminator codon is often called open reading frame or referred to as ORF. Within open reading frame, there is no scope for any codon to be non-coding, but there can be missense codons. If the reading frame changes by one or more nucleotides by insertion or deletion, the overall meaning of the message changes. The 10th point, in the 5' region, there may be several initiator codons, that is AUGs, very rarely GUGs, but only one of the AUGs will be in the right context in the sense the sequence around the initiator codon is important for correct and proper initiation of translation. If it initiates at the wrong AUG, translation will be terminated, for it certainly encounters a terminator codon. In the next point, in the case of prokaryotes, the coding region is split consisting of cistrons with initiating and terminator sequences. In between two such coding regions, one finds a short spacer region, hence the name polycistronic. In the 12th point, we have this spacer region is used for the translation termination of the cistron and again initiates the next cistron. The size of the intercistronic space varies, but mostly very short. Next, let us discuss about the features of eukaryotic mRNAs. The first point, all mRNAs are single-stranded polynucleotide chains with 5' to 3' polarity and mostly monocistronic. In the second point, the open reading frame starts with an initiator codon and ends with a terminator codon. In the third point, upstream of the initiator codon, pre-mRNA contains non-translatable sequences called leader or untranslated region UTR. Similarly, at the 3' end, after TER, that is a termination codon, most of the mRNAs contains another 3' untranslated region or 3' UTR trailer sequence which vary in length and sequence. In the fourth point, secondary structure in leader and 3' terminal regions plays an important role in the regulation of mRNA stability or its ability and efficiency to translate. The said 3' sequence are used for making the mRNA inactive to be stored for a long time. As for example, unfertilized eggs developing oocytes and plant seeds do store mRNA as informosomes. In the next point, the initiator codon should be in proper context for proper initiation. In eukaryotes, ACC, AUG, G or A or G, CC, AUG or G sequence is the most important sequence context without which translation initiation fails even if initiates. It will be terminated shortly for it encounters a termination codon. It is this sequence that acts at the time translation initiation is considered as the start of open reading frame. The said sequence is called Kozak sequence. In the sixth point, in addition to initiator sequences, positioning the Kozak sequence in the right context, the succeeding sequences have to be in the order 3 and should end in a terminator codon. It is only then such sequence is called open reading frame. Otherwise, if one finds one or more initiator codons in the 5' terminal region, one cannot consider it as a start of the reading frame. Any changes in this module makes the mRNA useless. In the seventh point, the 5' end of the mRNA is added with 7' methylguanine covalently to the first nucleotide, invariantly to the adenine residue by an unusual 5' phosphate to 5' phosphate linkage called capping. Subsequent addition of more methyl groups to 7' adenyl or second hydroxyl group of ribose sugars in succeeding nucleotides make them to be called cap1 or cap2, etc. In the eighth point, At the 3' end, mRNAs are added with a 200 to 250 nucleotide long polyadenine tail. However, eukaryotic mitochondrial mRNAs, as for example from yeast and neurospora, the poly-A tail is just 50 nucleotides long. In the 9th point, the poly-A signal with AAA, UAA and G or U rich sequence is used for cleaving the extended 3' end of mRNA at the 28 to 30 nucleotides from the signal. Then poly-A's are added to the cut ends. In the 10th point, the same mRNA produced in different tissues can be spliced in different ways by what is called alternate splicing to produce different polypeptides in stage-specific or tissue-specific manner, depending on the needs. In the next point we have, some mRNAs are edited in tissue-specific manner by specific enzymes or editosomes. In the next point, in eukaryotes, certain species of mRNAs are positioned in specific locations inside the cell. For example, segmentation mRNAs in developing Drosophila larvae, mRNAs for egg polarity fixation, and actins in developing myoblast cells. In the next point, such localization is made possible by their poly-A tails, where such mRNAs are actually held by cross-linking to actin filament networks. At the 14th point, the 5' untranslated region, it contains stem loop structures that have regulatory functions. Some of the mRNA contains internal ribosome entry elements called IREs. Next, the 3' untranslated region contains many sequence elements such as antisense binding sequences, regulatory stem loops, ARE sequences, mRNA localization sequences, and cytoplasmic polyadenylation elements. Next, let us discuss about transfer RNA, or commonly referred to as tRNA. tRNA has a clover leaf model structure leading to a three-dimensional conformation of the molecule. It has internal intra-strand hydrogen bonding that stabilizes the structure. The 3' terminal of the tRNA molecule is called the acceptor arm and has a conserved sequence of CCA that accepts amino acids. It has an anticodon loop that is complementary to the codon present on the mRNA. The anticodon loop is responsible for bringing the right amino acid at the translational unit during the process of polypeptide synthesis in the ribosome. Transfer RNA serves as the nucleic acid decoding device that reads the triplet genetic code of messenger RNA and causes the insertion of codon-specific amino acids in a growing protein chain during the process of translation in the ribosome. A particular triplet codon in an mRNA is read by a tRNA through its anticodon loop, which includes a triplet of anticodon residues that base pairs with the codon. Some of the modified bases present in a tRNA molecule are ribothymidine, inosine, pseudouridine, methylguanosine. Although there are 61 codons in the universal genetic code that specify amino acids, most organisms possess fewer than 45 different tRNAs. In continuation, we will be discussing about ribosomal RNA in the next topic. Ribosomal RNA or rRNA are RNA molecules that makes up the ribosomes in prokaryotes and eukaryotes. Bacteria and archaebacteria possess three distinct ribosomal RNAs, sometimes referred to as the 5S, 16S, and 23S forms. The 16S subunit makes up the 30S of the smaller ribosomal unit, and the 23S and 5S makes up the 50S subunit of the ribosome. In eukaryotes, where there is 80S type of ribosome, which has a 60S larger subunit and a 40S smaller subunit, there are smaller subunits of 5S, 5.8S, 18S, and 28S. The 5.8S and the 5S are homologous to the 5S of bacteria and archaeobacteria. The 18S is homologous to the 16S and the 28S is homologous to the 23S ribosomal subunit. The rRNA molecules in the ribosome folds over onto complex three-dimensional shapes. The specific shape that they take is highly conserved between species. However, the linear sequence of the nucleotide in each rRNA can vary without affecting the function, since different primary sequences can fold into the same shape. In conclusion, we have learned about different forms of RNA and their salient features. We have also discussed about differences between prokaryotic and eukaryotic mRNA. It is evident that RNA is the molecule that brings out the genetic information coded in the DNA into functional forms. RNA molecules are intermediary in the process of expression of genes, or in broad term, in the central dogma of molecular biology.